Good morning, everyone. Welcome to this virtual RSS meeting, jointly organised by the Environmental Statistics and the Emerging Technologies sections of the RSS. So we obviously, since you've registered for the meeting, you have noticed that we have an amazing lineup of speakers with different expertise in the field, and their talks will cover different challenges and opportunities of eDNA. We hope that the talks will generate interesting discussions between all of us, so we've allowed for plenty of time in between talks and at the end of the meeting, so we can all engage in interesting conversations and exchange ideas. You can ask questions by making comments in the chat during the talk, and then the chair will communicate these with the speakers. Or you can raise your hand, and after each talk, the chair will then invite you to ask questions directly to the speakers. As you've seen, probably your Microsoft Teams has let you know that the meeting is being recorded. So if you want us to delete your comments or questions before releasing the recording, please let us know. So before we start the meeting, I would like to remind you to obviously mute your microphone and turn off your camera. And I think we are now ready to start the meeting. Our first speaker is Kerry Walsh. She's a research scientist at for the Environment Agency, and she will be discussing with us the challenges and opportunities of eDNA from a regulator's perspective. So off to you, Kerry.
Thank you, Eleni. Can you just confirm that and you can hear me okay?
Yeah, it looks perfect and I can hear you very well. Thank you.
Great, thank you. And thank you for the invite today. So my name's Kerry Walsh and I sit within the Climate Change and Resource Efficiency Research team within the Environment Agency, where I lead on their DNA work programme. My talk today is probably the only non-technical talk of the day, and I apologise in advance that it doesn't contain any statistics either. But our interest in DNA-based methods started probably about a decade ago when we realised their potential application to our routine monitoring programme. And our work today is very much focused on freshwater systems. So today I'm going to give a brief introduction to what we do at the Environment Agency, sort of highlight what we've learned about DNA-based technologies and some of the challenges as regulators we're facing when we're thinking about implementing this technology into our monitoring programs to inform decision making and managing the environment, and then discuss some opportunities and future priorities. So although my focus is on the Environment Agency, I think many of the challenges are fundamental to all government organisations across the DEFRA agencies and the devolved administrations who are thinking of applying this technology. So all government agencies across the DEFRA group, so this is England only, work towards achieving the commitments that are set out in the 25-year environment plan. This is the government's high-level vision and commitment to improving the environment, and it sort of sets out how we'll achieve, for example, clean air and water, how we're going to have an environment that's richer in plants and wildlife, how we're going to be more sustainable and efficient with resources, and really importantly at the moment, how we're going to mitigate and adapt to climate change. And also this year, the Environment Agency published their 5-year action plan, and this sets out our contribution to the 25-year environment plan and other government commitments. Our focus really is on preparing for extreme weather events, meeting or surpassing the 2050 net zero commitments to improve air and water quality, to cut waste and improve public health. And DNA-based technologies, I think, have got real potential to benefit our work around air, around land, and around water. But it's the water environment that I'm going to focus on today. And the final document on this slide is an outcome indicator framework. And this sets out the indicators that the government will use to report on progress of that 25-year plan. And each indicator draws on a number of different metrics and data sets that are derived from different monitoring programs and other sources. Most of the metrics with an ecological basis use traditional ecological assessment methods. So there's lots of opportunities in this document where new DNA-based metrics could be developed to support the indicators outlined in this document, particularly around tracking changes in ecosystem function at a catchment scale in rivers and wetlands, and just generally tracking the state of wildlife that contribute to important ecosystem functions are just sort of a couple of examples. So where do we monitor? Well, a lot of our work focuses on protecting and improving the aquatic environment. And as part of that duty, we monitor the quality of rivers, lakes, groundwater, estuaries and coasts on a regular basis. A lot of our monitoring information is for statutory reporting under EU legislation, such as the Water Framework Directive and the Marine Strategy Framework Directive. A big focus of our DNA work has been on methods that support ecological monitoring under the Water Framework Directive in rivers and in lakes. So what sort of information do we collect routinely? Well, for the Water Framework Directive, we collect information on the physical habitat. So, for example, the natural features associated with rivers and any physical alterations to the channel or the banks. We collect a suite of water chemistry, such as phosphorus, nitrates, ammonia, dissolved oxygen. And in order to assess the ecological status of a given water body, we monitor the ecology. So we collect information on the presence and absence of single species, and we also collect community composition and relative abundance data on a suite of indicator organisms. And these include fish, macroinvertebrates, phytoplankton, benthic algae, such as diatoms, and macrophytes. So why do we monitor? Well, the methods that we currently use for ecological assessment are based on traditional biodiversity metrics and the pressure-specific biological indices. So the methods are used to understand the degree of impact of a pressure in relation to an unimpacted site, which we call a reference site. And this informs whether or not interventions need to be put in place to manage that pressure. And you can see some examples on the slide of the different pressures that we manage. So just to give you an example of what I mean by pressure specific, the indices that we use to measure diatom quality, for instance, in rivers is linked to eutrophication pressures. And this is assumed to be caused by phosphorus. So an indication of poor quality would require a measure to reduce phosphorus. Now, this is a really simplistic view of a really complex aquatic system where the quality of that system is actually governed by multiple pressures. And so therefore, our current approach presents a really restrictive approach to management, and actually it could lead to ineffective management and restoration measures put in place. So these pressure-specific indices, they provide only a limited amount of information on that bigger picture at the ecosystem level, despite the fact that we've got an increase in need to take more of a holistic approach. So taking a holistic approach at scale will give us much better insights into ecosystem health and resilience and the ways in which they function and continue to provide the services that, you know, that we all sort of now depend on. So much of our single species work is around the detection and management of invasive species such as the quagga mussel, signal crayfish, topmouth gudgeon are a few examples, and also protected species such as the freshwater pearl mussel and the white-clawed crayfish and the Great Crested Newt, which everybody is aware of. So our current ecological methods are very labour intensive, and they require a lot of time in the field, and then this is coupled to many hours in the lab looking down a microscope. But also for some taxonomic groups, such as fish in standing waters, some of the netting methods are actually harmful. So one of the main problems with our current methods is their high labour costs. The resolution is really variable across taxonomic groups, and assessment has been restricted to certain taxa that we can see, but actually these taxonomic groups may not be the best indicators of environmental change, and their cost, the cost of the traditional methods, make it difficult to upscale them in both time and space. So this slide just shows our average yearly costs, which is around 5 million just for 5 taxonomic groups in freshwater systems. And this doesn't reflect all our ecological monitoring commitments, but it's a huge resource commitment. So our interest in DNA initially started when we thought that it would bring efficiency savings, but then actually we realised that there was a much wider benefits, and that DNA technology had so much more potential to provide much deeper insight into understanding the environment much better. So our work's focused on both proof of concept work and full-scale development of methods using both bulk DNA and eDNA, and we've assessed metabarcoding for community-based assessment, and we've assessed qPCR for single species assessment. The potential of DNA-based technologies, as it's just raised such high expectations in the Environment Agency and across the DEFRA group in general, and we're really keen to start implementing these methods and putting them into operational use. So probably at this point, it's worth defining what we mean by bulk DNA and eDNA. And so in the context that we've used it, bulk DNA is DNA extracted from the isolated organism. So a species detected in a bulk sample was present at that location at the time the sample was taken. And an example of this is when we go out, we take a macroinvertebrate kick sample, we blend it, and we extract the DNA from that blended soup. And in contrast, eDNA is the DNA of an organism that's left behind in the environment. So it could have been transported from the original site of release. And this includes DNA from skin, saliva, feces, and urine. The development work that we've done has been, and that we've funded, has been supported through really good collaborations and networks that we've built via some initiatives. And I just want to highlight this because I think there's a real need for greater integration of data scientists and statisticians to be embedded into some of these initiatives. So in 2014, we created a DNA network, and this composed of government agencies, NGOs, academics, consultancies. And at the time, government agencies were just duplicating effort. There was no coordination of activities around DNA. So the aim of the network was really to provide a platform for better communication with each other, share learning, explore the technical challenges, and develop collaborative opportunities, and try and leverage research funding to facilitate developing these methods. The initial meeting attracted about 60 participants from the UK and overseas, and each year this has just grown, and interest has more than doubled. And the network now, it's got more of a formal status and governance under the UK Earth Observation Framework. And so for those interested, you can find out more about that at going to the UK EOF website. And in 2016, the EU cost initiative, the DNA Aquanet, was established. And this brought together, again, researchers across disciplines and other stakeholders. And the focus was really on developing DNA-based indices and metrics for biodiversity assessment under the Water Framework Directive. And 5 working groups were established to achieve their goals. And this initiative has got close links with the UK DNA network, and also with the more recently established the DEFRA Centre of Excellence for DNA. So last year, Natural England and the Environment Agency, we put forward a business case to DEFRA to establish a Centre of Excellence for DNA. This is now fully supported by the Chief Scientific Advisor, and it's got a series of projects running. And the aim of it is to speed up implementation of these methods into operational use and try and address some of the bottlenecks to their implementation. So what have we learned through our funded work? These are just some of the outputs of the published work, but essentially I think we sort of address 4 questions. So the first question was, can eDNA be used to detect invasive species? Invasive species assessment is really important because they're the second biggest threat to biodiversity after habitat loss. Both eDNA metabarcoding and PCR outperform traditional KitNet assessments when we were surveying invasive mussel species in rivers. And in addition, we found metabarcoding was really powerful for species discovery because we detected a native shrimp that's actually been around in our rivers for decades, but we'd never actually resolved it using microscopy. So our second question was about metabarcoding. Can DNA metabarcoding produce comparable community composition profiles compared to our traditional surveys? And in lakes, eDNA metabarcoding was comparable, and it was better when we were surveying chironomid midges and fish. But in contrast, eDNA from metabarcoding communities in rivers produce very different profiles compared to bulk DNA and traditional surveys. And I think this highlights the importance of eDNA dynamics and that consideration of waters. And our third question was, can DNA metabarcoding be used to determine the relative abundance of species? So when we looked at bulk and eDNA, we found correlations between sequence reads and biomass or abundance was quite variable. So for example, we found fish eDNA revealed significant correlations with sequence reads for both biomass and abundance when we compared it to absolute fish counts when we drained down ponds. We took advantage of a topmouth gudgeon eradication to look at absolute fish counts. Using mock community bulk DNA from macroinvertebrates, correlations between sequence read counts and biomass varied between species. And comparisons of DNA metabarcoding with shotgun sequencing on macroinvertebrate mock communities. So the shotgun sequencing removes that PCR step. We got highly significant correlations between read numbers and biomass. So I think it's generally accepted that the ability to quantify species biomass and relative abundance from metabarcoding, and whether that's eDNA or bulk DNA, remains a challenge, and I think an important challenge. And then our final question was to determine whether we could use DNA metabarcoding. Could we translate the data that comes out of that into metrics that we could use for reporting under the Water Framework Directive? And I think the answer is definitely yes. So we've developed new DNA-based metabarcoding methods and metrics for fish in lakes and diatoms in rivers. The fish method is soon to be implemented. It's been through consultation, but the diatom method hasn't. There's clear improvements that we could make to the diatom metric, and I think a lesson learned from our work was that we tried to develop a direct analogue of the traditional light microscopy metric. And in doing that, we brought over all the uncertainties in the original method into the new DNA method. And so by developing analogues, you fail to maximise all the information that's locked up in the DNA data. And so for diatoms, I think we need to re-explore the data sets that we've got using new statistical methods and try and develop a totally new metric for measuring status. So we know that as well as the huge potential around DNA-based methods, both of single species and multiple species, detections are prone to various sources of uncertainty and error. And errors can be introduced from contamination along the workflow. So from foreign eDNA that's introduced in the field, and that could be via wildlife or samplers, and cross-contamination between sites and between samples during processing, leading to false positive detection. Now, what makes eDNA so powerful is its sensitivity, to detect really low concentrations of eDNA, but then the trade-off makes it more susceptible to contamination. Suboptimal survey design and inefficient sampling and replication can lead to errors and species present not being detected and leading to false negatives. And then there's multiple sources of error that arise throughout the laboratory process and the bioinformatics, leading to misidentification and the creation of false positives and negatives. So such as, primers potentially not specific enough, bias in PCR amplification, so certain taxa are over-amplified compared to others, sample-to-sample contamination, and the quality of the reference database. So you've got multiple sources of error within a workflow, and for eDNA, this is then coupled to a lack of understanding about eDNA dynamics. So how much DNA is released from an organism? How long is it retained at that site before it's then transported downstream and degraded? The data produced from these workflows then needs ecological interpretation. We've got to put some ecological interpretation around that. And although we can mitigate some of these errors by modifying and optimizing sampling protocols, laboratory protocols, putting good QA and QC procedures in place, all of these uncertainties have manifested into organisational challenges. And the biggest is skepticism and confidence in the technology and the data that it produces. And this has been a real challenge within the organisation. I think a lack of standardisation in protocols hasn't helped either. So although traditional methods have many underlying uncertainties, and I challenge our ecologists and say this, but then they come back and say, yes, but we understand them, so we're able to work with them. And it's that lack of understanding around the uncertainties associated in particular with eDNA-based outputs, that drives the lack of confidence and skepticism. And there's a constant need to want to back that DNA data up with traditional surveys. I think part of the problem is that we've got a lack of capability and people with technical expertise within our organisation. And we know that there's a need to upskill our ecologists just to enable them to engage better with the technology. But aside from the formal training, I do think that knowledge exchange and engagement with the research community is key here. And this is where the DNA initiatives that bring people together really valuable platforms in our DNA journey. But another thing that I think is important is that the knowledge exchange has to be pitched at a level that's understandable to both policymakers and practitioners. You need to take them on that journey, because if we're serious about embedding this technology into government decision making to manage the environment, they need to have confidence and they need to be taken along, in that journey. So For the Environment Agency, I think we've got two key priorities. The first thing that we need to do is to address our current monitoring approach, because if we don't address that, then moving to DNA-based methods they'll have limited value in managing pressures. So we want to move away from our current single pressure, locally focused WFD monitoring program to a new surveillance network, and this is actually happening. We've developed a new river surveillance network that will be expanded to include lakes. It's based on a GRTS spatially balanced survey design, and this was originally developed by the US EPA, and Marine Scotland actually use it as part of their salmon surveillance program. It's a new long-term river monitoring program for England, but at the moment it's going to use Water Framework Directive compliant assessment methods, so diatoms, macroinvertebrates, macrophytes. It won't include fish initially because they're too expensive and it's the cost. It'll collect data on water quality, biology, morphology, and all of that data will be co-located, which is not what happens at the moment. So with our normal monitoring program, the biology and the chemistry monitoring points can be quite some distance apart, and this isn't ideal. And the scale of the new network at the moment is restricted by cost as it's using traditional methods, and so there's huge potential here for developing new DNA-based methods and metrics, and also I think, and this is what I'm keen on, is looking at new taxonomic groups that don't currently feature in our monitoring programs, but may be important indicators in trying to understand how our systems are functioning. And this is a shout out to sort of, the microorganisms. I think this is something that we should be looking at. The new system is designed to provide a framework for selecting monitoring sites that are statistically stronger than our current approach. And this is to improve the confidence that we've got in the data that we're then making decisions on. It's going to be scalable, so we can extract information at different scales. So we could look at river basin districts down to stream order, and also look at different land uses. The design, it's supposed to be representative of the whole river network, providing unbiased surveillance of status and trends at that broad scale. And I think the information that we're going to get out of the network is going to be very dependent on what we monitor and the statistical methods that we use to extract the information from that data. So we could extract very simple information, such as the status of a particular river, with respect to its macroinvertebrate community and chemical concentration, or we could look at try and extract more complex information using more sophisticated statistics to try and tease out information on that pressure response and what environmental covariates are driving that response. So initially, 500 sites per year will be monitored, and 100 of these will be fixed and visited each year, and the remainder of the sites will be selected from these different panels, and each panel is supposed to be representative of the population of rivers and streams across the river network in England. So the number of sites will be built on each year, increasing spatial representation, and the proportion of sites visited each year will increase to improve temporal confidence. And then after 5 years, 1,600 unique sites will have been visited. We hope the new network is going to be implemented in January 2021, and that new DNA-based methods could be incorporated into this as and when they become available. So the second priority is to address the technical challenges so that we can speed up implementation of these techniques, and I've listed a few that I think are important to us. I appreciate there's a lot of work happening within the DNA research community to improve methods, to standardise approaches, and also to improve our understanding of eDNA dynamics in aquatic systems. What we haven't seen much of is the actual translation of metabarcoding data into new metrics that we can use to manage the environment. And I think there's many opportunities for new DNA-based metrics to be developed to support government priorities. And that outcome indicator framework document that I put up initially is where, you know, there are metrics needed to support managing the environment. So we know that we can produce good quality data, high resolution data on ecological community composition. However, because community composition and relative abundance are key components in metrics that we use to understand the impacts of change, we need to understand, can we generate more accurate abundance estimates from DNA? Can we do this statistically or in the laboratory, or do we need to move away from metabarcoding to a different technology that, you know, that doesn't rely on PCR? Improving the reference database is really important, and curating them to improve quality. And this is actually a priority under the DEFRA Centre of Excellence. We're working in collaboration with the Natural History Museum here to try and improve the reference database for key UK taxa. And then there's the developing new statistical methods based on DNA data so that we can quantify uncertainties in metabarcoding workflows. I know new models are out there for single species data, and this will certainly start to improve confidence in using that data. Optimizing sampling designs that balance sensitivity and cost. They need to be realistic and they've got to be pragmatic. Otherwise, you know, we just won't implement them. And I think this is an area that's not really received the attention that it deserves and would benefit from, you know, statistician involvement early on in the method development process rather than at the end. Understanding complex interactions and identifying drivers of change. How can we tease apart the key pressures on ecological communities from the data? I think this is a real key priority for us. And then the final one is really from, you know, it's come from questions around our invasive species work about using eDNA, and although and building on the growing body of evidence that we're getting on eDNA dynamics, is it going to be possible to infer species location from eDNA data? And I think that is a really important question. And finally, I've been to 7 conferences of the UK DNA Network since 2014, and I've seen some fantastic work presented on, you know, DNA-based technologies, what they can do and deliver with respect to biodiversity assessment, how it can help us understand and manage the environment. I've seen how the technology outperforms traditional methods in almost all cases. I've seen the DNA research community improve methodologies. They're working towards standardization to improve confidence. But last year, at the conference last year, it was the first time that I've heard statisticians talk about how they're contributing to this area. And I think this was exciting, because I think we are at a point now where there needs to be much better integration of different disciplines if DNA-based methods, you know, if we're going to realise their true potential, and for stakeholders to have confidence to use and implement them. And I think that's me. Thank you.
Oh, thank you very much, Kerry. What a fantastic talk to kickstart the event. It's really highlighted basically exactly that, the challenges and the opportunities that eDNA offers. Is there, if people want to ask any questions, please pop them in the chat, or you can raise your hand, and then I will invite you to unmute yourself. But while people are kind of warming up to ask their questions, so would you say, so obviously you've mentioned a lot that you've compared the results you get from, for example, metabarcoding compared to your traditional surveys, and would you say that you think that you would be looking more towards metabarcoding rather than, say, single species PCR, or do you think that each has its merits depending on what what you want to monitor?
It's very much about the remit and the questions that you're asking. So I think they are both important, but the community, you know, for us, we've got such a large monitoring program in terms of understanding community structure, so composition and relative abundance, that metabarcoding is definitely key. Or any other technology that gives you, you know, information on community structure.
And just keeping an eye on the chat as well at the same time. So, I mean, you already mentioned one of the issues, for example, is the reference libraries when it comes to metabarcoding. But how are we in the UK? Are we in a fairly good position in terms of kind of having up to date and as complete as possible reference libraries? You mentioned you were working with the Natural History Museum on that.
Yeah. Well, DNA Aquanet have also looked at gaps in reference databases, but that was sort of on a European scale. And the work we did was looking at UK species, you know, specifically, and identifying what species we currently use within our monitoring programs to prioritise them. And I think, you know, most of the European databases, I'm not 100% sure on the figure, but I think it was something about 4%. You know, the European and more international reference databases contain something, you know, about 4% of UK taxa. And I know from our experience, you know, we developed a diatom reference database, and that cost us about £100,000. And we actually only had 10% coverage of UK diatoms, you know, having invested, you know, £100,000 into the barcode reference database. So I think in terms of UK taxa, we've got a lot to do, but it is definitely up on the Centre of Excellence, you know, priority list.
Oh, so we've got a question from Nick. Is the RSN linking into the Darwin Tree of Life project to sequence a reference organism from every species in the UK? This should massively improve our reference databases. Maybe more of a comment than a question combined.
So
thank you, Nick.
So actually, the Centre of Excellence is liaising with the Darwin Tree of Life, and because that is heavily linked to the Natural History Museum as well. So, and that's why we are wanting to sort of, you know, put forward key species that, you know, that we want priority in terms of, you know, generating barcodes that are actually important in our ecological assessment methods. So that is happening.
Okay. Are there any other questions? Someone's raised their hand. So, Laurie, do you have a question to ask? Laurie.
Are you muted, Laurie?
Oh, yes, you're muted still. Yeah.
Still can't hear
you. Did you just have to press the little if you no, it doesn't. Do you want to type it in the chat and I can read it out? It says one more question from Chris. If we have particular interest ideas about the specific areas you have mentioned, can you suggest ways we, the statisticians, that was going to be my next question actually, can contribute to those conversations? For example, Chris says he works on optimal sampling design, but not in the context of eDNA yet.
I think get in touch with me, and you know, and then we take it sort of from there. But I think it's not just about the Environment Agency. I think, there needs to be that integrate. There's lots of DNA work going on with Natural England, Forest Research, the Environment Agency, and through the Centre of Excellence. So I think the DEFRA Centre of Excellence would, is a good route into that, because we sort of oversee the UK DNA network. There's close links to all of that. So, but I definitely say, next year's UK DNA Network conference was supposed to have been in January, but it's looking like it's potentially going to be March, or even later next year. And I definitely sort of encourage, statisticians coming to these conferences and building the networks and getting a good understanding of what's actually going on.
Oh, absolutely. I can get. I mean, I encourage everyone. I attended last year for the first time. The Natural History Museum, beautiful venue and very interesting conference. I've got one more question from David from CFAS that I've missed. So excited by the new river surveillance network, but did you say won't include fish? Also, will it link up to the site of the National River Flows Archive, as flows an important area?
I haven't been involved in the development, so I can't say what it links up to. At the moment, fish aren't included, and this is because it's a cost-driven decision at the moment, and this is why we need DNA tools to start integrating. Fish is one of our biggest resource burdens, and it's the most costly. So at the moment, it's just going to be the suite of WFD methods minus fish. But if people are really interested in the RSN network, the person in the Environment Agency that has designed all that and is really involved in the statistics side is more than happy to link up with people to give them more information about it, how it's going to work. Because even though we're not using DNA-based methods, even looking at new statistical methods and models to deal with the data, the traditional data that we've got, I'm sure that she'd want to sort of link up with people.
Yeah, that's really good. Thank you so much. So I've got a couple more questions. So I think it's fine because we've got a couple more minutes. So Nick's come back. Do you see a role for unselected methods, example WGS, I don't know what that stands for, or methods without amplification in the future of eDNA surveillance?
I'm not sure I got the first part.
Yeah, I don't know what, maybe Nick can explain what unselective methods, he says, example WGS.
Whole genome sequencing, I'm assuming.
Oh, it's probably, yeah.
Oh, definitely. I mean, we want the methods that are going to provide the best data outputs, but we can ask for that, but then there's a cost associated with that, because some of the newer methods are more costly. So it's I think there's trade-offs to be had here. Do we balance specificity of a method, sensitivity of a method with cost, and I think we do that with traditional methods, that they aren't perfect. So in response to it, it depends what data these unselected methods are going to deliver, and whether that type of data is the type of data that we use as part of our management decisions. And if we don't, then tell us how we can use it.
Okay. And then so Laurie says she's not muted. Must be a problem. I'll check later. Sorry. So fantastic talk, Kerry. Thank you. She completely agrees about engaging statisticians in the design and early stages of projects. She also wanted to pick up on the point you made about water chemistry. We need to work with chemists and hydrologists to better understand DNA dynamics. Having access to EA data and capability when collecting eDNA samples would be really helpful. Environment Agency.
Yeah.
Smiley face.
Well, there's a problem with our data, I think, and I don't know, I'm not a statistician, whether or not statistically you can account for, you know, biology and chemistry in a water body being, you know, quite a distance apart when you're then trying to infer, you know, an impact of a pressure. But we used our own data as part of developing the diatom metric, and in hindsight we should have collected co-located data, and also there's some issues with detection limits for things like, you know, phosphorus. So yeah, I'm interested. We've got a huge data set for diatoms. We've got 1,500 samples of matched light microscopy and next generation sequencing data, with associated environmental chemistry data, and you know, we can go to the CEH site and get flow data and things like that. I'm really keen to get data science people, statisticians, to try and explore this data further, and actually to look at our water chemistry data to, you know, to see if it is valuable or not.
That's excellent. I'm so pleased with how amazing the talk was and how interesting the discussion was afterwards. Thank you so much, Kerry, for this amazing talk. So our next speaker now is Naomi Ewald. So she's a Director of Policy and Research at the Freshwater Habitats Trust, and she will be presenting about how eDNA data can inform conservation priorities, and in particular using case studies of long-term species monitoring and short-term before-after surveys. So off to you, Naomi. I can see your slides, and I can hear you very well.
Brilliant, that's lovely. Thank you very much. So I'm going to go from really one extreme to the other, following on from Kerry's presentation, working for a small NGO charity called Freshwater Habitats Trust. And we are very much practitioners working in the field to undertake practical conservation work in order to really protect, enhance and increase freshwater biodiversity in the UK. So the opportunity to have a technique which could help us with our work, obviously when eDNA was first came onto the scene, was really significant for us. So I'm going to talk about a single species, Great Crested Newts, and a lot of you on here will know quite a lot about them and will have seen what we've done with them to date, but we still have quite a lot of challenges in terms of analysing the data and working out where we go with it next. And that's where I'm hoping you will come in at the end with a whole load of questions for me. So just stepping back a little bit and giving you an overview. For us, what's really important is that small water bodies make up the vast majority of the freshwater network. So more than 90% of the freshwater network is made up of small standing water bodies. And globally, around 10% of all species are found in fresh waters in just 2 to 3% of the land surface. So we've got a large resource and a lot of biodiversity in that resource. However, they are actually, particularly in the water body, protected. Work we have done over the last 30 years as an organisation has identified two really critical things. Firstly, that 90% of ponds in the UK are probably in a degraded state. So they're unmonitored and degraded, but where they are high quality and where you create high quality sites, you can increase gamma diversity around 25% by just putting in a small number of ponds into the landscape. So we stand in this place where we know there's a lot of water out there. We don't know what's in a lot of that water. We know that some of that water is still high quality. Most of it isn't high quality. And we know that if we can get it right, we can make a big difference. So just in terms of what do we monitor at the moment in the way of species, individuals, so that we have a really good idea about the status and change of those species, I think most people would argue and say that currently most species aren't very well represented, except for birds and butterflies in the UK, which have a very long history of monitoring and understanding about how their populations are responding to change. In terms of freshwater species, salmon and sea trout are well monitored for economic reasons. Otter are well monitored, although not annually. Natterjack toad are really well monitored, but they're not very representative of the wider countryside, even though most people who work with natterjack toad will be able to tell you what the names of each individual are. And then for other species, we had the National Amphibian and Reptile Recording Scheme, which was really a first attempt at recording amphibians and reptiles at a national scale. That was a volunteer-run programme, and it was very difficult to get volunteers to complete the required numbers of sites. So whilst it was very successful for engagement and very good at gathering data which had not been collected previously, it's not been very useful in terms of an ongoing monitoring tool. And similarly, the Dragonfly Recording Scheme has been great at gathering data, but there's still not enough experts out there who are able to undertake the surveying to be able to say something about change over time. And surprisingly, until we undertook this project, there really wasn't any good monitoring data, even for highly protected species like Great Crested Newts. They are a European protected species, and as part of Article 17 reporting, every 6 years the requirement was to for member states to respond on what the status of Great Crested Newts was in the countries where they occur. And for the UK, we were unable to come up with an answer because the monitoring data wasn't there, in spite of the fact that millions of pounds are spent on Great Crested Newt every year because of the requirements for undertaking developments and really protecting every individual newt as a protected species. So in spite of that, we still didn't have enough data to be able to say something about their status. So it's very hard to know what's happening to freshwater species and why. It's very difficult to know what we can do about it. So as I said, Great Crested Newts are a European protected species and are very controversial as a species because they are at the forefront of what people see as an obstacle to development going forward. And so they have quite a high profile. But until recently, before eDNA, there was only a limited number of techniques in order to identify them. These are obviously cryptic species, they're nocturnal, they're very secretive, and even in sites where you know they occur, you can undertake a huge amount of survey work and not encounter a newt when you're at the site. So traditionally, the techniques that we used to survey them were time-consuming and required both skilled and licensed surveyors. And many of you probably already know this, but the traditional methods are bottle trapping, where you set bottles, basically empty bottles, on canes overnight, and the newt's very inquisitive and they go to those bottles and then can be recorded the next morning. You can look at them with shining a torch into the pond. You can undertake egg searching in leaves, provided you are skilled enough to be able to identify those eggs. And you can undertake basic netting where you fish around in the hope that you might find one. And work that has been done, and it's very detailed work, to look at the effectiveness of those techniques and also look at really whether you are detecting them or not. So whether an absence is a true absence. You have to undertake 4 visits using each of those techniques in order to have 90% certainty that you have detected newts when they are there. And if we think about that in terms of costs, these traditional surveys require 2 surveyors who are trained and skilled, so their costs are high. Undertaking 4 visits for each pond, you could be looking at between £1,600 and £2,400 per pond. There's a very short period of time when you're able to do this in the breeding season. And so to undertake a national survey to record newts presence in ponds would be prohibitively expensive to do it using consultants. And the alternative is to train up volunteers and give them a site to go out and survey. But again, at a national level, volunteers get very jaded if they keep being sent out to ponds that have no newts in them. The first time you clamber over a field fence and walk across in the dark is exciting. The second, third, and then fourth time becomes slightly less interesting. And so this was often the problem with the NARS surveys. The surveys weren't completed. So we had a lot of missing gaps in the data, and we were only getting results back from ponds where it was almost certain that newts would occur. So that's not really giving you a good impression. So in 2013, we were approached, it was just before Christmas and New Year, by the DEFRA family to see whether this new technique, eDNA, would be useful for helping them to look at the status and change of Great Crested Newts in the UK. And I'll come back to this at the end, but just to say, we undertook a very detailed programme of assessing the technique, seeing whether it could be applied in the field, and then setting up a volunteer monitoring network to use it. From the beginning to actually rolling it out took us 3 months. And then from rolling it out to actually setting up the national programme took another 8 months. So I think sometimes there is a perception that these testing, trialling and coming up with an understanding of what the limits of these techniques are takes a long time. But for us, particularly with the single species surveys, it's incredibly quick. So what I'm just going to talk through is the 4 areas of work that we went through to get to this stage. The first one was developing and testing the primer for Great Crested Newts in the UK, then putting in place a programme of field testing to evaluate any factors that might affect the eDNA detectability in the field, so false positives, false negatives, carrying out then some statistical analysis comparing the eDNA to traditional techniques, and then producing practical guidance on what sort of sampling methodology we might need at a national scale to monitor them. So developing the primer was fairly straightforward, and those of you that have been developing eDNA will be very familiar with this. We already had GCN primers, but we wanted to test the probes that we were using within the laboratory, and found that the probes were perfectly aligned with the Great Crested Newt, so cristatus being the scientific name for those of you that don't know that, but there were also some mismatches. So whilst it was perfect for that species, we were getting then some mismatch with other species. So then we went out into the field and collected some newt swabs of known Great Crested and took those back to test them in the lab again against these probes and against the primers, and again found that whilst it was able to pick up Great Crested Newts, we were also picking up some overlap with 2 other of the species of the same family. Fortunately for us, those at that stage had not been recorded in the UK, so it was felt that that would be an acceptable error that we would deal with going forward. We then, as Kerry mentioned, were looking at the limit of detection, because obviously we were trying to find very small amounts of neat DNA in very large literal pools of water. We undertook that analysis, and this was working with Spygen, who are a French laboratory who had done a lot of work with amphibians in Europe, and so had a really good track record, and were very confident that they would be able to provide us with sensible limit of detection and limit of quantification, and then looked at how many replicates we would need to do of each. So this laboratory stage of it was fairly straightforward. They were looking therefore, what the errors would be at that analytical stage. And I think from what we have seen going forward in terms of the metabarcoding and other single species work, this element of it is really now pretty tried and trusted. So we've got a very good idea of how these kits perform in a laboratory setting. For us as practitioners, what we are interested in is the errors that are generated in the field, both the sort of natural elements, but also the surveyor elements that can really make a difference to your results. So this was the next stage, to undertake a field test, trying to develop detailed and standardized techniques to limit any errors that we might encounter in the field. So the first thing we did was to compare eDNA techniques with standard methods. So the torching, the bottle trapping, the egg searching. We didn't use the netting technique because actually even in traditional surveys that is considered to be a poor technique and only used as a last resort. So we concentrated on the first three techniques. We also felt that netting at a pond where you were trying to have some balance along with eDNA would require us to stir up the ponds too much. So we didn't test that technique. We looked at 35 ponds in total across 2 different locations in the UK, so North Wales and Hampshire. And we undertook the 4 recommended visits, late April to late June. All of these ponds were known to support good populations of Great Crested Newts in advance. So here we were really just comparing the method. And so the results here, you can see that the eDNA performed extremely well. So looking at the graph in the top left-hand corner, first of all, eDNA, the number of ponds in which Great Crested Newts were detected by each survey method on each of the 4 visits. And for eDNA, it was 99.3% detection rate. And that translates to only 1 visit at 1 pond where we were not able to pick up newts being there. That was an extremely shallow pond, was very boggy to get to, and almost certainly that was surveyor error and not kit error that meant that we had that non-detection. But the point of this part of the survey was to compare it to the other methods. And you can see on their own, for bottle trapping, 76% detection, for torch counts, 75% detection, and for egg searching, 44% detection. So eDNA is outstripping all of the techniques that traditionally have been used on every single visit. So rather than doing the 4 visits, you could get the same level of detection with just 1 visit. And because that's slightly unfair comparison, because in traditional techniques you would never just use one of those methods, it's always a combination of methods, and usually it's torch counting alongside bottle trapping. So we combined the results of the 2 traditional methods and again compared it with the eDNA, and you can see that even when you combine the 2, you get 95% detection using combined methods, eDNA 99% detection, so they are comparable across each of those visits. The main difference, obviously, is that eDNA is much quicker in the field to do the torch counting and the bottle trapping at each of those sites on 4 visits. We're talking here of a difference between 2 hours for eDNA and 48 hours for the traditional method. So really important results. And in terms of selling the concept of eDNA to other practitioners and also to reassure government that we were able to reliably detect the species using this method, understanding what the error was part of the story. This gave them the confidence to actually then back rolling this out more into a national programme. So that was part element of the survey was done using consultants, and we were very confident that they were skilled. And so the next challenge was then to roll it out and see whether we could get similar results using a volunteer task force to undertake this national monitoring. So trying it on a large scale, we had 86 volunteers and they went out to 239 sites, all known for Great Crested Newts. And we were looking there at assessing the kits in terms of was there any variation in their ability to detect newts depending on the pond type, and could volunteers practically use the technique. And the answer was a very definite yes. Of those ponds that were known for Great Crested Newt, 91.2% of the time, newts were detected using eDNA. So again, that's performing similar levels to how traditional techniques would have performed, only you would never have been able to test that many sites using volunteers using traditional techniques. Where we were getting false negatives, in other words, the newts were there but the kits weren't picking them up, there were really two main issues. One was the pond type. If the ponds were very difficult to access, volunteers really struggled to be able to get a good water sample. We were recommending taking 20 samples from around the whole perimeter of the pond, evenly spaced, to get a really good sample up to ponds of 1 hectare in size. So access made a huge difference to the result. But also we were picking up that in ponds that were very turbid, either because of sediment runoff or because of turbidity caused by fish or, for example, geese, then the ability of the kit to pick out the newt DNA, even when newts were there, was limited. So that again caused a few false negatives. And on a very few sites, we were clearly reaching the limit of detection for the kits. So if there were known to be only 1 or 2 newts present in the pond, which could have just been individuals passing through, again, the kits weren't picking them up. So overall, a successful rollout to test whether volunteers could undertake the survey, but again, knowing the limit of the kit. And I think what's happened since is, again, there's this thing that Kerry was mentioning around confidence of the kits. People have said, oh, well, yes, I've been out and used these kits on a pond. I know there are newts there and I'm getting a negative result back. So these kits are rubbish. And I think that's because there is, again, a misunderstanding that no technique is ever going to be 100% effective. So I think we need to get better at helping people to understand what the error rate is and what that means for their results. So we also undertook, as final part of this, a test to see whether we got false positives, in other words, from ponds where no newts had ever occurred, and then seeing whether the kits gave a false reading. So we went to sites outside of the range of Great Crested Newts in Cornwall and Shetland. They all came back negative, and we went to ponds within Great Crested Newt range, but where newts had never been recorded. And again, they all came back negative. So we had 100% success in getting no false positives, but some false negatives. And this has all been written up into a paper, which I hope these slides will be circulated, so you'll be able to look at that. Okay, so that's the first part of the talk and gets us to a point at the end of 2013 where we felt very confident that eDNA was going to be the solution, and we would, for the first time in the world, be able to set up the first amphibian monitoring network using eDNA to gather reliable, robust results on the status and change of this species. But the question was, how many sites do we need to visit in order to then to be able to say something about status and change? Newts are complicated for not just the fact that they're hard to survey when even in sites where you know they are, but the distribution across the country is uneven, and they occur in very few sites. So if you just undertake a random survey, most of the ponds you visit, you won't find any Great Crested Newts. So you could waste a huge amount of resources just getting a lot of negative results. And then even once you've got some positive results, if that is just a handful of sites, it's really not enough to tell you anything significant. So, you know, obviously what we're talking about here is understanding the sample size and seeing what we needed to build in terms of this national network. So we undertook a power analysis. And to start off with, in terms of resources, we thought that government was only going to be able to fund us to do this survey twice. So with a 6-year gap. So do it at time point 1, wait 6 years, time point 2, and see whether we had got a significant change between those 2 time points. And so undertaking that kind of design, we estimated that you would need to survey around 550 1 kilometre grid squares, because that is the unit of measurement that we are interested in reporting on for Great Crested Newt occupancy. So how many 1 kilometre grid squares are they occurring at in the UK, and what's the change over time. But even at 550 squares, we were only saying 80% power to pick up a 30% change in GCN occupancy. And for us, a 30% change in the occurrence of this species is a big change. If they are over a 6-year period declining by 30%, then we're in trouble. If they're increasing by 30%, everyone would be terribly shocked and it would be really good news. And this as well was based on a stratified sample where we had sort of split them into 2 categories. So 50% of the squares were known to have previous records of Great Crested Newts, so that we would have proportional representation in the sample of enough sites with known presence, and then 50% unknown, so that we could get a better handle on what the real distribution status of newts was in the UK, because we didn't have that information before. And then also, hopefully over time, if conservation measures are effective, we would see expansion into those areas. So that was based, the power analysis, on very sketchy data. We pooled records from environmental record centres, from some consultancy records, from some sites, a very small number of sites, 5 in fact, where we had some long-term monitoring of what was happening to Great Crested Newts over time in those places using traditional techniques. And so I would say this was really almost a finger in the air exercise. We put some data in, we got a result out, true results or whether it was just what the information we had, which was terribly biased information. So still 550 is ambitious as a volunteer network if you want them to go to all of the ponds within each 1 kilometre grid square. The second issue that was then raised was that DEFRA didn't have enough money for us to survey 551 kilometre grid squares, even using this new technique without having all of the costs of consultancies. And in fact, they only had enough funding for 100 1 kilometre grid squares, so about 300 ponds in total. And we managed to get a bit of extra funding to do 131 1 kilometre grid squares, 380 ponds in total. But the trade-off was that they said that rather than doing it as a one-off survey repeated every 6 years, they would fund an annual survey over 6 years. So we would have 6 years worth of data, and we are just in the 6th year now. So the results are being processed at the moment and we haven't had them back. So what I'm going to present to you is just the first 5 years. Okay, so again, I'm going to step back and just give you a little bit of information in the hope that you're all going to come up at the end of this with some brilliant statistical solutions to allowing us to analyse these data in a better way. So there are a number of ways to monitor Great Crested Newt at a national level. I've put here a typical 1 kilometre grid square. We can aim to survey all of the ponds in that 1 kilometre grid square to give us lots of pieces of information. With that, we would be able to say whether that square is occupied or not, full stop, because if it occurs in any one of those ponds, then the square is occupied. We can look at the number of occupied ponds by taking a pond as representative within each 1 kilometre grid square and then saying, okay, across the network, what is the pond occupancy? And we can look at the number of occupied ponds per square. And so our setup was designed to say, yes, we'll try and monitor all of the ponds in each 1 kilometre grid square, and allowing for the fact that many landowners will refuse permission and some eventualities may mean that we lose the ability to access the pond. So rather than aiming for every single pond, we said we would aim for 75% of the ponds in each 1 kilometre grid square, but then that network would become fixed. So over the 6 years, we would always revisit the same squares and the same ponds. And so this is what the results look like so far. You can see that in terms of square occupancy, so the number of occupied squares in England, for the first time, we were able to report that occupancy is around 25% of 1 kilometre grid squares, and that stays relatively even across the 6 years. So actually we've detected no change over that time period in square occupancy. There was a slight increase in 2018, which I'll talk about in a second. It was a good year for newts. It was a very mild, wet summer. Newts tend to have core ponds, which they're in every year without fail, and then satellite ponds that they will move into to breed in what they are obviously perceiving as a good year, and then they will retract back to their core ponds in poor years, which is why we ended up with 2017, a peak in 2018, and then dropping back down in 2019. The issue here is that this occupancy is almost certainly bouncing along at the bottom of the potential for newts in the UK. Historically, they probably would have occupied around 80 to 90% of ponds, and they've declined dramatically over the last 50 to 100 years. So we're really picking up here a very impoverished population. And so these are the ones that are clinging on, probably in optimal habitat where they have retreated to. And so that's one of the reasons we're maybe not seeing the change. 6 years also is 5 years in this graph is a very short amount of time. We wouldn't necessarily expect to see change over that time. And so the question is, how long do we need to keep doing this survey before we have a chance of saying something about changing Great Crested Newt population? And secondly, are we already surveying enough sites? Is this correct to say that we're actually not detecting a change, or are we just failing to survey enough places? In terms of pond occupancy then, so looking at the pond level rather than at the square level, you can see that pond occupancy didn't change very much over the first 3 years of the survey, sticking at around 13%. This was higher than previous estimates, so eDNA is certainly detecting smaller populations than had been previously detected using traditional methods. We got that peak in 2018, and then it's dipped back down slightly 2019, still no change statistically over this time. One of the other issues that was thrown into the mix during this time is that between 2017 and 2018, we had to change laboratories for the analysis of the kits. So we had an issue about whether this peak in 2018 was caused because the lab was better at detecting them than the previous lab. But when they went back down in 2019, we felt that maybe that was less of an issue, but there hasn't really been significant interlab testing so that, again, we can quantify what's happening here. And it makes a real difference when you're trying to discuss the conservation of a single species to really understand what your results are showing. So we've gone from here to saying, OK, we've now got 6 years of data. How much longer do we need to keep doing this? Can we have a break for a few years and then pick it up again, or should we keep going? I obviously want to keep going. And so we've done a bit of analysis, and this is where, in terms of the statisticians we're using at the moment, we've got some questions, and I'd be really keen for you guys to feed into it. So taking this trend data of occupancy in 1 kilometre grid squares over time, so this is just binary, positive, negative, as to whether they're there or not, and how that is changing over the 6 years. We used the long power package in R to undertake sample size calculations for this longitudinal data, using the presence of GCN as response variable, and then having a continuous fixed effect variable of year within each site. And at the moment, what we are being told, although again, I think there are probably better ways of analysing this, is that if we specify that we're looking for levels of change of between 5, 10 and 20% in occupancy over X number of years, and again looking at a level of power between 60 and 90%, and saying whether we're going to do 2 time points or continue with the continuous survey. Monitoring annually over 6 years and accounting for the variation. At the moment, the sample size we have of 131 1 kilometre grid squares theoretically is allowing us to pick up 10% change in square occupancy over that time period, with around 80 to 95% power. And for the policymakers, that is something that they have a lot of confidence in. They are very happy with a technique that can pick up 10% change in newt populations over 6 years with the sort of level of costs involved of surveying around 100 to 150 ponds. And they have used that for favourable status conservation reporting or favourable species conservation reporting for the Article 17, and are now rolling it out to actually use it as part of district level licensing to determine what offsetting and compensation developers should make in order to undertake development where newts are present. However, the newts have behaved so strangely and created so much variability in the number of years that we've been looking at, probably in response to changes in precipitation and temperature in those years, that there are a lot of covariables that we haven't yet included in the model. And I would like someone with far more knowledge to help us undertake that part of the analysis. So very quickly, I'm just going to finish by talking about another element of this, which is where we're going next. As many of you will know, the single species eDNA uses the qPCR technique, and there is now a lot of talk about whether we should be using the metabarcoding technique instead to actually get more information out of our samples than just great crested newts on their own. So it seems a shame to go all that way to collect a sample and then not get the other information. And so as part of the district level licensing, which, as I said, is the technique which is now being used to decide what compensation there should be for development, we reanalysed eDNA samples taken with qPCR using metabarcoding to see what the difference was between the two techniques for this species and for other species. So you can see here is a map of ponds across the South Midlands where we are using the technique for district level licensing, and presence and absence of different species. So for the first time, the map on the left, we got data about other amphibians which we previously didn't have about these ponds. So clearly using metabarcoding is useful. On the right hand side is the great crested newts to say, well, did this technique perform as well as the single species eDNA? And actually we found that it wasn't as effective. So the metabarcoding was only able to detect great crested newt in about 2/3 of the ponds where we knew that they occurred. So again, if we are going to roll out this technique and replace the single species technique with multi-species technique, we would need to understand again what the error rate was between those techniques so that we again can give the policymakers confidence about what we're reporting on. Okay, particularly for great crested newts, one of the advantages of using the metabarcoding is its ability to detect fish, because that's one of the biggest drivers of the presence or absence of great crested newt. And so again, completely unachievable previously, but at the moment, because of the lack of in-depth testing about what these results are really showing you compared to what the reality is, it's quite difficult to then just switch technique and roll that out with confidence. And very finally, for us as an organisation, because we are there trying to work and conserve these species on the ground, we are actually very keen to develop single species eDNA kits for a wider range of lesser known and difficult to survey species which have conservation status, particularly so that we can survey more sites, so that we can get more people out there collecting this information, and so that we can then kickstart the sort of policies that have driven the development of the Great Crested Newt eDNA technique for these other species. Okay, thank you very much.
Thank you so much, Naomi. What an amazing and interesting talk. It really highlights as you said, the potential for especially qPCR compared to traditional methods that rely on direct observation of the species. And it's it's amazing what you guys have done. And as you said, it's really early on, so already in 2014, where our statisticians weren't even aware that eDNA was a thing. So that's excellent. So there are a few questions, actually quite a few questions. Chat is on fire. So Nick has a couple of quite technical questions. So Nick, if you don't mind, I might give Naomi a chance to have a look at those after I've asked my question. So maybe during the break.
Sure.
So Andrew says, so he has a question about the limit of detection for the primer. Does this identify a threshold for the amount of DNA and then assume that detectability is perfect above this threshold? How is this threshold chosen? So I guess Andrew's referring to the amplification where you interested in whether a more flexible model could be useful here. Smooth function, that model's detectability is a function of amount of DNA.
Yeah, I think so. So yeah, so it was a very crude analysis, basically with a dilution factor, and then we just said, okay, yes, below that we're saying that it's not able to detect it, but above it we assumed 100% at that stage. Again, because this was such early days in the development of this, it was good just to have something rather than nothing. So you're right, there is more work that is obviously being done now, but we haven't then included that in our model. So we haven't in the models that we're doing and the change over time and the presence or absence of Great Crested Newt, we're not actually at this stage building in anything about detectability either in the field or in the lab, and that again is a gap in the analysis that we would like to build in, because that will help with the reliability of what we're concluding at the end of it.
Thank you. So Laurie has another question. Sorry, Laurie, another. I mean, you tried to ask a question for Kerry as well, but so I haven't heard back from Luz about your microphone not unmuting. So she says, thanks for a great talk. You may be familiar with a paper titled Needle in a Haystack, a comparison of eDNA metabarcoding and targeted qPCR for detection of the Great Crested Newt. It's a paper in Ecology and Evolution from 2018.
Yes, I am. Yes.
Yes.
And this, so at the moment, this is a really interesting area, because we've got a couple of things going on here. Is I would say 3 areas of work that we're trying to look at. Firstly is as we've just talked about, whether to go from a single species to metabarcoding techniques. And as you say, there's a lot of work being done on that, and we're just wondering now if we're at the stage where we can move across and say with some confidence, yes, we're going to do that because it makes sense. But we need to then have a very strong argument to be able to change that, because this is part of the policy planning. The second one is whether we can accept, even now, moving from an alcohol-based kit, which is what the original kits were, to a filter kit. So a change in the actual design of the kits. The filter kits are better in terms of detectability. We've seen that we actually get better detectability using that kit, even if we're then still using the qPCR technique. The actual kit itself seems to be better. But again, because we're looking at change over time, if we then suddenly have more detectability, what does that do to our analysis? We would have to account for that in the model. And then thirdly, still this issue around the reliability of between lab comparison, and having a better idea about the error around that. So I think where this technique has moved on so quickly, but we've begun a journey looking at trend over time, we're at that point now where do we, after 6 years, update the methods and then carry on and then have some ability to analyse across the board?
Yeah, that's actually a very good point, because if you have a consistent error, then you can always talk about relative changes, right? Whereas if you, as you say, if you change the way you do things, then you have to account for these changes. And I think Doug had exactly that point you've just made about using filtering instead of ethanol in your samples. I think that you've just covered that.
Yeah, we would love to, because for the volunteers, it would make the process so much easier. That it would transform and bring the costs down actually quite substantially.
Okay, that's really interesting. So Andrew again says lots of work on detectability up here in St Andrews, so could potentially future work on that.
Great.
Laurie says she definitely agrees with Doug, but it's not a completely fair comparison. 12 replicates of qPCR versus 1, 2, 3 for metabarcoding typically and broad range primers. Yeah, I was going to ask about that actually, because so from the work we've done, you probably are aware of the models that we've been developing and that Jim will be presenting later on this afternoon, where we estimate the probabilities of observation error in both stages. And we found that clearly people tend to collect a single water sample, so you don't have replication at the site level. So when you try and estimate the probability of observation error, false positive, negative, you don't have basically that much data because you just have that single observation. It's like, if you're anything where you're trying to estimate probability of something happening, you want to try something a few times, right? And then you do the 12 qPCRs, and obviously then in stage 2, that gives you a lot more power to say anything about observation error. So is that because of cost that people tend to collect? So you have done the same, a single water sample from each?
Yes, that's right. We did. We have experimented and sometimes taken more samples. And when we started this with SpyGen, we did do a piece of work around that where we were actually taking 5 samples sometimes, but only because of costs, we only did that at about 3 sites. And the results all came back the same. If the newts were there, then all the kits detected them, and if the newts weren't there, then none of the kits detected them. And that wasn't really enough then to justify doing more work around that. But there is a question where we're seeing the differences between years on some ponds. Is that because there's a very low population there? Is that picking up the population leaving, or is it the kit, or do we need more replicates at some sites in order to find those very small populations?
Yeah, and I think your talk again highlighted the need for a more informed study design, so as did Kerry's talk. So I hope the statisticians in the field get it. So hope I'm not. So the chat is really on fire. So Helen, I think I'm not missing anything. Helen says just a question on the filters, so not totally related to the talk. Trying to order Sterivex filters today, only to be told that they have been discontinued. Has anyone else come across this?
I haven't, but I'm not at the forefront of the kit ordering. So if anyone else on the chat wants to chip in.
Oh yeah, yes, someone's replied. Yeah. So, right, sorry. So then another question from Nick: is a single large water sample different from taking smaller separate samples? He doesn't think so. I also don't think so. I think that's definitely quite a discussion we've been having here at Kent, also with Richard and Andy, about how, because do you collect various samples and then mix them all together in a bag?
Yeah, so we try to get a representative sample. So the 20 points, and I think there was another question that was coming up about that. So the 20 points that we collect around the pond are evenly spread out to try and get access, you know, to the whole water body. But then we do homogenize that sample within the Steripack and then take then the pipette from that into the 6 vials, which are then analysed back at the lab. So that's how we're doing it. So in effect, it's just one sample from the pond, but we try to make it a good sample. And we did find that where you have ponds with limited access, that was one of the issues in detectability, that if you weren't able to get 20 samples all the way around the pond, in other words, get a good single sample, it was very easy to miss newts, even if they were in there at quite a high density.
That's really good. Okay, so Eric agrees with Helen about the same problem with Sterivex. And so David asks, 20 points. Yeah, that's the one. I think that they're just like how now there's a discussion going on between the, the participants. Okay, so I think if there's another question that is related directly to this talk, we might just break here. So Nick's asking a couple of technical questions on the actual statistics. So for example, about your histograms and what the bars represent, and in terms of percentage change. So I don't know if you want to have a think, go back to your calculations to try and answer the question. So if there's obviously you can take part in the chat, either about Sterivex or about standard deviations. Thank you very much, Naomi. That was an excellent talk, and again, lots of stimulating discussions. So we'll break until 30 past, so 11:30, and we'll come back with Francesca Picciola for the last morning talk. All right, everyone. I hope you had a bit of a break away from your screen. So our third speaker for this morning, so it's the last morning talk before break for lunch, is by Francesco Picciola. Francesco is a professor at the Department of Environmental Science and Policy at the University of Milan, and he'll be presenting the use of eDNA when tracking long-term changes of mountain ecosystems. Francesco, if you're there, you're going to share your screen, and whenever you're ready, we can start.
Sure. Thank you very much. It's a pleasure.
Thank you for giving this talk.
Okay, can you see the screen?
Yes, we can see it, and we can see you, and we can hear you really well.
Excellent. So thank you very much. I'm happy to be here, and I'm going to talk about a very different system, but I hope to show you that mountain environments are another very interesting system to study the relationships between human activities and environmental changes, and can provide a very interesting idea on more things we can do to use environmental DNA to understand our impact on ecosystems. So the mountain environments have been inhabited by humans since a lot of time, and there are already evident impacts by humans since the Neolithic. And this is evident from the Alps, where I work a lot, but it's actually evident in mountain ranges all around the world. And of course, through time, humans had different impacts on the ecosystems. There are different processes that drive the changes of these environments, and we know now that we have a strong impact of climate changes, but we also have direct impact of human activities. You have the impact of biotic interactions, and of course there are natural processes like the geological processes that occur through time and also concur to determine the long-term changes of these ecosystems. So what I will try to show you here is an example of approaches that can be used to reconstruct the long-term interactions between humans and the mountain environments, and try to measure how human activities can affect the dynamics and, at the end, the functioning of mountain environments, and essentially understand how those dynamics influence the biodiversity. And we are discussing about environmental DNA, and here I will mostly focus on analysis that have been performed using metabarcoding, and Noemi already described well this approach, so I'm not telling about how does it work. But in any case, I will show you how this can be used to identify a very, very broad range of organisms. So the first example I'm showing you is related to the use of sediment cores taken from lakes to reconstruct the long-term changes of the environments through time. So here we are at a special scale finer than what we have seen so far, while we are going to look in a long-term temporal scale. So a lake gather the sediments that come from the catchment around the lake. So the erosion brings sediments from the mountains inside the lake, and a sediment is essentially a portion of soil that is transported from the mountains to the lake. And year by year, we have the deposits of sediments accumulate through time, and actually the paleoecologists are using this kind of sediments since a lot of time to reconstruct environmental changes. Because, for example, we can reconstruct the past soil, we can obtain paleoclimatic reconstructions, we can obtain pollens from sediment cores, and in the last years we discovered that we can also extract environmental DNA from the lake cores. So I'm going to show you a couple of examples. This one is from the Alps, on how the environmental DNA from the lake cores can help us to understand what happened in the past, how the environments changed in the past, and why do we see a given environment today. For example, this is the Antern Lake, it's in the French Alps, and here you see what is called an alpine meadow. It's a lake at about 2,000 meters of altitude. So if you ask people, why do you have a meadow here? Why there are no trees? Why there is no forest here? And most of people will answer, well, of course, we are at the 2,000 meters of altitude, so it's too cold for forests, and so you only have the grass. And alpine lakes are nice places to go, and you can go there and collect sediment cores, both during summer or during winter. In that case, you have you can put big cores, and you can obtain very, very long sediment cores that can go very deep in the past. And so you can take sediment samples that represent different periods. So you go deep in the past by collecting a sediment sample, and you can extract the environmental DNA from those sediment samples. And this is an example from this core, and the core was pretty long. So we have been able to extract ancient DNA up to about 7,000 years ago. And it's possible to find the DNA of many organisms, both mammals and plants. Those are the organisms we targeted in that specific study. And for example, if you look at the DNA of mammals, you see that most of the mammals you detect are domestic mammals, mostly cattle and sheep. And the first detections of cattle DNA date back to 5,000 years ago, but most of the DNA of domestic mammals, so mostly cattle and sheep, starts since the Roman period, so about a little bit more than 2,000 years ago. If you look at the vegetation, you see something that is very different from what you can see today, because in fact, from 6 to 3,000 years ago, you find a quite frequent presence of the DNA of pines. And this suggests that you had trees that actually are not present today. And in the moment of about 2,000 years ago, you lose the presence of pines, and there are other plant species that appear. And the pines are not alone, because with the pine, you also have more plant species that are typical of alpine high altitude forests. So you have not just the trees, but also the herbaceous plants that live below the pines, the typical vegetation of a forest. More recently, around 2,000 years ago, you see something completely different. So the pines disappear, and also the plants that are associated to the pine forest disappear, and you have a very different situation with the grasses that live that are associated to the alpine grasslands we see now. So this suggests that the arrival of mammals, of domestic mammals that have been kept by farmers since the Roman period has determined a very, very strong change of the environment. And what we see now is not a natural grassland. Natural alpine grassland probably occur at higher altitudes. So we have evidence, for example, from other lakes that about 500 meters above this lake, we always had grassland through time. But in this point is actually the effect of the human activities, the presence of domestic mammals that determined the changes that occurred not today, not a few years ago, but we still have the long-term consequences of environmental changes that occurred about 2,000 years ago. I'll show you a second example from in principle, very different system. And again, to show you how the interaction between human activities or the indirect effects of human activities can have very long-term effects on the ecosystems. And again, using environmental DNA, we can obtain, in this case, a quite high resolution picture of what happened in the past. So we are going very far away from Europe. This is Kerguelen Island. So Kerguelen Island is one of the most isolated places on the planet. You see, is halfway between South Africa and the Antarctic. So it's in the middle of the southern Indian Ocean, and of course it's a quite cold place. And if you look again at the landscape, you say, oh, it's very, very desert landscape. There is not a lot of vegetation, and we can assume that we have this kind of very desert landscape because of the climate, because not many plants can grow in such a cold climate. And the island is one of the less human inhabited places in the world. There is it's an island that has several hundred square kilometers of surface, so it's not small at all. But there is about 10 people living there. There is only a scientific station to study the mostly the climate and the geological events of the area. But even in those very isolated, lonely places, we have animals that have been introduced by humans. In Kerguelen, we have rabbits, and of course, rabbits are native of Europe, but have been introduced by humans about 150 years ago, because in 1864, a French expedition for the study of planet, where astronomers went to Kerguelen and spent several weeks there, and they had also rabbits that were of course used as a food. But at the end of the expedition, when the scientists left the island, they also left a few rabbits, I think 5. So in case somebody was coming back to the island, some food would have been available for people coming. And we now know that rabbits graze a lot on native vegetation, and specifically in this native plant, which is called the azorella. And but actually, we see that now the rabbits graze on azorella, but we don't know what is the real impact of the rabbits, and if the scarcity of azorella in the landscape is really a consequence of the arrival of the rabbits. And also we have no idea about how fast is the interaction between the rabbits and the vegetation loss that we can assume occurred. So again, there is a picture of Kerguelen. You can see it's not a small island, and but like many other places in cold climates, you have plenty of lakes, and the water is cold. This is good for the preservation of environmental DNA. So in this case, we took a sediment core from a small lake in the middle of the island, more or less, and we know that the rabbits have been introduced here in 1860, and we know that now rabbits are around in the landscape, but we have no idea about when the rabbits arrived. And we have no idea, of course, because nobody lives there, on how the interactions between the rabbits and the native vegetation occurred through time. Remember, this is a place where there is only a very small scientific station here, where about 10 people live, and nobody else all around the island. So again, in this case, we extracted the environmental DNA from the sediment core, and here I show you the DNA of 2 species of plants, Azorella and Acina. Both are native plants. And so the core covered about 1,000 years. So far in the past, So, for example, in 1380, Azorella was the most abundant plant species. This plot shows you the relative abundance as proportion of eDNA reads between Azorella and Acina. So in the past, 700 years, 600 years ago, or 500 years ago, Azorella was the dominant plant. And also about 150 years ago, Azorella was more or less stable and a little bit more abundant than Acina. Acina is in orange. But then you see something completely different. In the sediment core that dates back to 1945, you have a very strong increase in the abundance of Azorella reeds. And in 1945, Azorella reeds are about 95% of all the environmental DNA you find in the core. And then it decreases through time, and now it's very, very small amount of the DNA. And this is exactly what we see in the landscape, because now Azorella is a rare plant in that landscape. So there is not so much Azorella in the landscape and not so much environmental DNA. So what did happen in 1945? If you look at the mammal DNA, you only find the DNA of one mammal species, no other mammals. Well, there are almost no other mammals in Kerguelen, and in any case, there are no native mammals. And of course, there is no DNA of rabbit far in the past. Rabbits were introduced in Kerguelen in around 1850, but it took time clearly to spread from the introduction point to the lake. And the first rabbit DNA was detected in 1945. And the first detection of rabbit DNA exactly corresponds to the year in which you have an increase of Azorella. And after the presence of rabbits increases, then you see the strong decline of Azorella, which essentially go disappearing. So what is the interpretation? Essentially, the interpretation is that when rabbits arrive, they graze mostly on Azorella, and the DNA you find is the DNA of Azorella that the erosion brings down to the lake, for example, because the DNA of Azorella, the remains of plants, are inside the feces of the rabbits. Or when rabbits excavate their burrows, they increase the erosion rate, and this again increases the amount of plant remains, plant roots, plant whatever that go down inside the lake. And there is an impressive match actually between the abundance of rabbit, in this case is estimated on the basis of spores of coprophilous fungi, and the erosion rate. You see that the arrival of rabbits exactly corresponds to the increase of erosion in the landscape. Then we see also a decline of rabbits through time, perhaps because of a disease that was introduced to the island, or perhaps because well, there is no more food, and so the populations declined. So this shows to you that we can track very well and very accurately the effects of environmental changes, in this case the introduction of an alien species, on the different aspects of the ecosystems using environmental DNA. And the last example I'm describing is this is an ongoing process. So it's a project that we started a few years ago and is on the present day and maybe future dynamics of mountain ecosystems in response to to climate change, and we are mostly focusing on the retreat of glaciers. So we all know that because of that climate change is ongoing, climate change is very fast, and mountains are among the environments where climate change has the strongest effects. So all around the world, you see glaciers that are retreating. And these are pictures taken from exactly the same point, and you see that glacier lost, this is one of the largest glaciers of the Alps, and you see how fast it lost its surface through time. But after a glacier disappears, life comes and it completely modifies the landscape. So a few years after the retreat of the glacier, you don't have, or you don't see actually a lot of life. You essentially have bare substrate, but quickly plants and animals and microorganisms colonize those sediments, and just 1 century after, you can see forests with 20 or 30-meter tall trees. And the research on the consequences of the retreat of glaciers is a very classical field of ecological research, but so far, using the traditional approaches, you have to identify the plants or the insects or maybe some clade of microorganisms using traditional approaches, and this takes a lot of time. And so far, most of studies have been performed on very, very small geographic areas, and it's very, very time-consuming to assess the presence of multiple organisms at the same time, because it would require many specialists working together. But the environmental DNA can be extracted from soil, and in the soil you find the DNA of all the organisms that interact with the soil. So of course you have the soil organisms, soil animals, but you also have the cells of plants, because plants have roots inside the soil, and plant material fell down, and the DNA is trapped inside the particles of the soil, and again you can find the DNA of microorganisms. So what we are trying is to extract DNA from those terrains where glaciers disappeared recently, and we are trying to reconstruct the whole community, and to understand how the functional diversity changes through time, so how the different functions of those ecosystems emerge. So we are, this is a very metabarcoding approach, in the sense that we are trying to target many different group of organisms. We use some markers that amplify many species, but have the drawback is that they have a limited resolution. So we have a marker that amplifies all the bacteria and archaeobacteria, one marker that amplifies all the eukaryotes, some markers that are specific to plants and fungi, and some more and more specific markers for the organisms that can have a particularly important functional role, like the springtails that in the soil are one of the main components of the food webs. So in the last years, we have been relatively lucky, because the project involves a global sampling of glaciers all around the world, and likely we almost finished the sampling before the coronavirus epidemics. And so we have been able to obtain samples from glaciers all around the world, from New Zealand to Canada. And it's good to know that glaciologists have excellent data on the retreat of glaciers in glaciers from all around the world. So this is an example from a glacier in Bolivia, where you can clearly see where the glacier was present in 1830, 1870, 1900, or 2005, and so on. And going around to look those glaciers sometimes gives you very, very impressive views of how fast climate change is. So this picture was taken 1 year ago, so you can see that this is a glacier in Iceland, and it lost 1 kilometer in just 9 years. And in any case, what we are trying is combining those the information I'm going to show you a little bit on the environmental DNA, with information that is available in time series that sometimes already exist, that have been collected using traditional approaches on the vegetation, on the arthropods, and of course on the modifications of the cryosphere. So we are performing analysis at 2 temporal scales. In most of glaciers, we are obtaining samples that go deep in the past, so from moraines essentially that are about 150 years older. So those are the most developed stages of colonization after the retreat of glaciers. But also for a number of glaciers, we are also trying to perform a high resolution reconstruction of what happens. So we go in the field, we collect soil samples that represent different ages after the glacial retreat, and we extract the environmental DNA from the soil, and using metabarcoding, we reconstruct the communities. And then we are combining this information on the communities with information on habitat modifications. So how soil changes, how occurs the development of soil in terms, for example, of organic matter, nutrients, and also we are combining this with a high resolution reconstruction of microclimate. And so what we are trying to see is, for example, what is the rate of colonization of the different organisms, who comes first, and how the first colonizers affect what happens at the subsequent stages. And we are trying to compare different models of colonization. For example, do we see a replacement of pioneer species with species that are more adapted to the later successional stages, or do we see the subsequent addition of more species? And I show you a couple of examples. This is an example from Charquini, it's a glacier in Bolivia. And here you can see that the patterns of biodiversity change are completely different if you look at different groups of organisms. If you look at the protists, so these are microscopic eukaryotes, the richness, the species richness, those are not really species, they are molecular taxonomic units, but the diversity don't increase through time. Actually, you have the highest diversity very, very early after the retreat of glaciers. This means that the protists are able to colonize this environment very, very quickly. While for animals and plants, you have a completely different pattern with an increase through time of the number of species you detect. And those changes in the number of taxa doesn't mean just you have more species, but it also means that you have more functions and the food webs, for example, increase in complexity. So this is again an example, another glacier in Bolivia. Very, very soon, just 8 years after the rise of glaciers, you already have animals, but you don't have many animals. We only detected the 3 taxonomic units, and the number of taxonomic units increases in older terrains, but you don't have just more taxonomic units. You have different functions. So at the beginning, you only have bacterivorous and decompositors. But things completely change through time, even if the 2 terrains are exactly the same altitude, more than 4,000 meters of altitude, and you have more functional groups. So you have the grazers, you have the parasites, you have the predators that were not present at the beginning, and no species is in common. You have 100% turnover. This means that you have different functions that start, and also that different organisms are adapted to the different stages of the development. Okay, this is what. So, and essentially, you have a much more complex food web. And what we are trying here is to combine the data from many different glaciers to have an overall picture of what is the response of biodiversity to this form of climate change in general. Okay. And thank you very much. And if you have questions, I'm here.
Thank you so much. What a fascinating talk. I can't believe how big and ambitious this project was. And you're right. You were lucky to manage to get all of your data in just before we all had to get locked inside our houses. It's just amazing to see how much you can now do, how far back in time you can go and learn about communities thousands of years ago. And I think in terms of statistics, you've got a lot going on there. As you say, you've got your time series, you've got change points. So where things are in a certain way and a certain model would apply, and then suddenly things swap, like you were showing us with the distribution of plants. So in terms of statistics, kind of you've got kind of extinction models that could also apply. So there's a lot of interesting statistical methods out there that would be really useful, where you can put in covariates and so on.
In terms of statistics, one of the main challenges we have are the false absences, because we have particularly if you go deep in the past, I'm sure there are plenty of organisms that were present in the past, but we are not able to find. For example, we have to increase really a lot number of replicates to reduce the number of taxa we don't detect, to reduce the false absences. This is a huge problem when you go deep in the past.
Yeah, so I see Erica had a question about this, but you've just answered that question, she says. Yeah, I mean, I'm not surprised. I mean, it was kind of, but when you were looking at the detection of, for example, cattle DNA, it was obvious that you had some blips and then nothing and then another. So that it wasn't consistent, that you could identify, oh, this is a period, and they were not there before then, they were not there after that. It's probably they were then between periods when you detected them, but it was just you didn't
no, absolutely, no, this is absolutely true. I didn't show you, but we actually we with a PhD student, we tried to combine this method with autoregressive methods, so with trying to limit this effect of spikes, that because I'm sure, just to give an example here, I don't have cattle detections, but I'm sure that cattle was present, maybe at lower abundance, or maybe just because I mean, in that study, we performed 8 replicates per sample, but in another study, I'm not showing you, but we arrived up to 24 replicates per sample to be as much confident as possible of the absence of the cattle. And this is this is really problematic. But there is definitely room to improve, because you have to filter another problem, which are the false positives, that are also possible and are particularly frequent when we don't have a lot of DNA. And in that case, because, for example, there is always a little bit of cattle DNA in the environment, because cattle are our main food source for humans. So even in the water, in the reactives you use during the PCRs, you always have a little bit of pig and cattle and maybe potato DNA. That's very, very challenging.
Yeah, absolutely. I mean, I can imagine. And so how many different, because you're targeting animals and plants and bacteria and so on. So from each sample that you collect from each point, how many different species across all of these different taxa maybe would you find, generally speaking?
It depends. It's very variable. Just to make clear, for example, for Kerguelen, the diversity of plants is very low.
Yeah.
And so you have 12 plant species. You cannot have more than 12 plant species. But when you go to the very small animals or the bacteria, you can have 100 of taxonomic units per single soil sample. You can find, I don't know, 10 species of earthworm and 5 or 10 of springtails, and hundreds, but really hundreds of fungi or of bacteria. And of course, you have the trade-off between the false positives and false negatives there also.
Yeah, no, absolutely. So Richard says, fascinating stuff. Any idea whether the eDNA signals are related to the hydrology or catchment area of the lake? So dilution effect.
Yeah, there are two different, there are different analyses that have been performed on this, and there are two different systems. I will call them alpine lakes. So you have streams that bring the sediments inside the lake. And in that case, having a good, well-developed drainage around the lake is good, because it brings more sediments inside and gives you a very good sedimentation rate. While in a study performed in Norway, and found a completely different pattern, because it was where small lakes in relatively flat landscape. In that case, we have a very, very narrow strip around the lake that provides the DNA from the surroundings to the lake. So you essentially found, for example, the DNA of the vegetation that grows a few meters from the lake and no more. But this depends on the processes that determine the sedimentation.
So that's, I mean, yeah, it's really mind blowing how much work you've done with this, and how much, I'm guessing you have identified so many interesting results, and that what you've shown us is just a small sample of what you guys have been kind of trying to put together, right?
Yeah, this is a couple. And each time you analyze a new lake, each lake tells you a different story, because it's a different ecosystems. But then when you put many lakes together, for example, actually we have analyzed about 15 different lakes from the Alps, and then we have a number of multiple lakes from the Alps. Then you see that there is a number of general patterns. For example, what I show you, what you see here. So the peak of domestic mammals in the Roman period, and the loss of native vegetation in the Roman period. This is absolutely widespread, at least in the Alps, and this is something that is general at the alpine level. But in any case, every lake has a different altitude and climate and composition of the vegetation community, and so the pattern is absolutely never identical.
That's really interesting. So if thank you so much for this amazing presentation. Really fascinating stuff. So if anyone, if there are no more questions, then I would say thank you once more. And we can break for lunch, I believe, that time of day. And we'll be back at 12:45. So we've got half an hour, so it's not a very long lunch break, but just enough maybe to stretch our legs, grab a bite, and we'll be back at 12:45 with Doug as our next speaker. Thank you very much.